coming to the invasive species set of talks here. What I want to talk about today, my name is Martin Bontrager. I'm from the University of Wisconsin-Madison. I work in Carol Lee's lab, and we work on a species of copepod that has invaded from saltwater to freshwater multiple times independently. And I want to talk about the bacterial community that's associated with that copepod as it transitions from saltwater to freshwater. So let me start with a little bit of background about copepods, for those of you who don't know. Copepods are estimated to be the largest metazoan biomass in the ocean. Despite that fact, they're very tiny. The species we work on is approximately 1 millimeter in size, or something like that. They are the single most important food source for many economically important fisheries, including haddock and smelt and flounder. So they are not only ecologically very important, but also economically important. Some previous work has shown that concentrations of bacteria associated with copepods in the water column are orders of magnitude higher than in comparable volumes of the water. So they seem to be a microbial hotspot in the water environment. And other work has shown that the copepod gut is an anoxic or hypoxic environment in the otherwise oxygenated water column that might provide a unique niche for bacteria to colonize. The species that we work on in our lab is called Eurytemera affinis. This is a dominant or the dominant copepod in coastal and estuarine environments throughout the northern hemisphere. This species is interesting because recently it has invaded from saltwater to freshwater multiple times independently from different ancestral source populations. And when I say recently, I mean within the last 50 to 100 years in most cases, so very recently. This has been documented, and on this figure, not to belabor the point because I'll go over it later, but we have 3 invasive lineages. One that I've colored red in the northeast US, one that I've colored green in the south, and then purple invasive lineage in Europe. These invasions are often accompanied by parallel evolutionary shifts from the saltwater to freshwater invasion. So the Lee Lab has documented shifts in salinity tolerance and nutrition requirements and gene expression in parallel in these independent invasions. These replicate invasions, these independence of these invasions, make this a powerful model to look at parallelism during invasion. So I started this project with a few broad questions that I just want to go over. First of all, we don't know much about what the bacterial community is like associated with the copepod. So our first question is, what is the taxonomic composition of that community? And is it different from the surrounding water? Is it unique from the environment that the copepod inhabits? What environmental factors structure the copepod microbiome? So we went into this thinking that salinity obviously might be a major environmental factor, but things like latitude, temperature, seasonality might be another important factor. And importantly for the purposes of invasions, what happens to this microbiome as the copepod moves from a saline environment to a freshwater environment? What shifts? Are those shifts that happen in parallel, independently, or are they unique shifts per clade? What could we tell about that? To address these questions, we used a sequencing approach. We sequenced the 16S ribosomal RNA gene of the copepod microbiome and of the surrounding water environment from multiple locations in Europe and North America. So I'll just briefly talk about this map. This is our sampling location map. From each of these locations, stay in this invasive red lineage, we collected 100 Eurytemera affinis, our species, and collected around 10 liters of water from those locations and isolated bacteria. And then we sequenced those bacterial communities to determine what was there. In this figure, you can see, like, for example, the dark red dots are saline environments, light red are fresh, and that's true in green and purple clades as well. That was our collection scheme. Once we sequenced, we could bin these sequences by identity. In this case, we've been at 97% identity at an approximate species level. So I'm going to call that an observational taxonomic unit, or OTU, which is to say that the 16S gene sequences that are 97% identical or higher are one OTU, or a proxy for species. So what happens when we compare the copepod microbiome with the surrounding water? First and most importantly, the copepod microbiomes are highly distinct from water microbiomes. So let me walk through this figure a little bit. You see there's 6 facets to this figure. Each one represents a different metric or different method of determining how similar different population or different microbiomes are from one another. It doesn't matter exactly what they are. The point is that in each of these 6 different distance metrics, the copepods represented by red dots and the water samples represented by blue dots cluster independently of one another along this first principal coordinate axis. So the similarities between these communities are being driven by whether they're host associated or whether they're in the water. They're clearly very distinct. That is highly significant via Permanova testing. Furthermore, water microbiomes are significantly more diverse than copepod microbiomes. So again, I'll direct your attention to this figure, and not to belabor the point too much, but let's look at the first facet, the observed taxa, that is the observed number of observational taxonomic units, or the observed number of species, let's say. There are significantly more species or OTUs associated with water microbiomes than there are with copepod microbiomes. In this case, the mean number of OTUs in a copepod is 290 about, and in the water is about 1,350. So that's just looking at presence-absence. These other 2 facets scale that diversity by community evenness, so the proportion of those bacteria. In either case, this is a highly significant result. The water is very diverse and the copepod is less so. Perhaps surprisingly, at least to me, there is no correlation between the diversity in the water and the diversity in the copepod microbiome. You may expect that since they come from the same location and they share a lot of environmental traits in common, like temperature and latitude, other things, you might expect that a higher diversity water sample would be correlated with a higher diversity copepod sample. That is not the case. So these 2 communities are actually very independent of one another, water and copepods. And so what is the copepod microbiome composed of? So in this figure, I've highlighted the top 10 phyla, bacterial phyla, that exist in all of our sampling. And you can see, and this is scaled by relative abundance on the y-axis, you can see here that water communities are dominated predominantly by the actinobacteria. This is this pink group of bacteria here. They're not absent, but almost absent from copepod microbiomes. So this is clearly something that the water has, the copepods do not. In addition, we find that the Firmicutes, which is a small brown bar there, but is almost absent from the water, are highly enriched in the copepod. This is interesting. Other groups have found this as well, that Firmicutes are highly enriched in zooplankton communities. So we find the same thing. That could be because of this property of the copepod gut being anoxic, or without oxygen, because many members of the Firmicutes are obligate anaerobes or facultative anaerobes. All of our copepod microbiomes contain one particular species of Ledbeterella, which is absent from all of our water microbiomes. I find that interesting that across this broad geographical range, this one particular species of bacteria or strain is present in everything, which certainly might warrant some follow-up on our part, but hasn't been done yet. This is just an interesting finding. Copepod microbiomes also contain several fish pathogens, known fish pathogens, which I might talk about later if I have time. So that's when we compare water microbiomes and copepod microbiomes, but what about when we compare the copepod bacterial communities with one another? This is the same type of plot, a non-metric multi-dimensional scaling plot, but think of it as a principal coordinate analysis plot of copepod microbiomes and their similarity to one another. And I've colored in dark red saltwater microbiomes and in light red fresh. So this is the only environmental factor that we found that actually structured similarity between communities in the copepod microbiome, and its salinity, and it's highly significant. So this is what is structuring the similarity between, the only environmental factor that we know of that is structuring similarity among these communities. That's highly significant by permanova as well. That clustering by salinity suggests some type of parallelism during a species invasion. Freshwater bacterial communities associated with copepods are always more similar to one another than they are to saline, or clustered together, more than they do with saline environments. And that's interesting, because in our data, when we look at specific taxa or even families, we don't really see that pattern. There are a few OTUs, a few species that are more abundant in the saltwater copepods versus fresh, and there are a few that are more abundant in freshwater versus salt, or actually there's one that I found. But really, this structuring by salinity is not driven by any one or even two bacterial taxa. It's being driven by an entire, the entire complement that are somehow more similar to one another. So I think that's kind of interesting. We also found quite a bit of evidence for clade specificity in our data. So in this red invasive clade, this northeastern clade in the US, there's a member of the Rickettsia genus. Rickettsia is an obligate intracellular symbiont that's present in a lot of arthropods. Specifically, it's been found in Drosophila and a lot of other beetles, but the reason I find it interesting is it often affects host fitness and sex determination. It's a very tightly linked symbiont of many arthropods. And I think that warrants a lot of future study because it is only in our northeastern population and is absent from the rest of our sampling. Those are true. There's other cases of that in our sampling. For example, in the European clade, there's a member of the Holospora, which is a intranuclear paramecium parasite. Not quite sure what that means yet, to be totally honest with you. But so anyway, we find this clade specificity as well in our data. So overall, what I want to impart to you is that ours is the first study to examine the microbiome of an aquatic invertebrate in this comparative context across like a fundamental biogeographic barrier, like the saltwater to freshwater barrier. We found that copepod microbiomes are more similar to one another than they are to the surrounding water. And perhaps the most interesting finding, at least to me, is that the salinity of the surrounding water really structures the copepod microbiomes, their similarity to one another. Also, we found that the water microbiome is significantly more diverse, or if you prefer, the copepod microbiome is not as diverse as water. And I won't belabor this due to time, but our future directions, or what we're kind of working on now, is looking at fish pathogens in these copepod microbiomes and how they're significantly enriched. For example, these three genera are major fish pathogen genera. Here's their abundance in water, and in the copepod microbiome, they're significantly enriched. We've actually isolated a species of Flavobacterium from copepods and infected fish with it, and it kills fish. So this is an interesting finding that points to copepods as a potential reservoir of fish diseases. I got to do a lot of fun work, which is basically the whole point of this slide, to show what I did, which is cool. And I want to thank everybody involved, including Jane Rumford, who sits there and there, who helped me with a lot of the sampling in this project, everybody in the Lee Lab, the Silva Lab at University of Maryland, and the NSF for funding a National Science Foundation Graduate Research Fellowship, and I'd be happy to take any questions.